I'm here to introduce quickly a person who is a Dreyfus Teacher Scholar in 1971, Professor Steve Lippard. And Steve has been chair of the MIT Chemistry Department for 10 years. Wow. Okay. He's also received the highest award from the American Chemical Society, the Priestley Medal. Steve loves New York City. I met Steve when I was with him on the faculty at Columbia University. Steve still does all his shopping for clothes in New York, I believe. And let's welcome him as he tells us about cancer and platinum.
Well, thanks very much, Dick, for that unusual introduction, which I appreciate. I still eat and buy my clothes in New York. You only gave half the statement, actually. So it's a great pleasure to be here. I want to congratulate all the teacher scholars for your achievements and having been awarded in this way. You will carry this with you forever, and I hope you will enjoy it and spend some time inspiring future teacher scholars to come. So, and thank you, Mark, and for the kind invitation, and also Jerry, who's done a great job in organizing everything. So my work is at the interface of inorganic chemistry and biology, and there are two major avenues in this field. One of them relates to understanding the existing inorganic species in biology, so metal catalysis and enzymes and so forth. And the other is the introduction of metals as probes and pharmaceuticals. And today I'm going to talk about our work on platinum anti-cancer drugs. And there'll be two sections of this. One is related to understanding how the existing drugs work, talking a little bit about how they were discovered, a story which I imagine some of you have heard already. And the other is to show you how we think we can improve the situation. And we'll do that in that order. So first of all, the discovery came in the hands of a man by the name of Barnett Rosenberg, who's a biophysicist. I'm sure many of you have heard this story, and I've related it a number of times, but I never get tired of telling it. So Barney, a physicist, was interested in the lines of force of electric fields and their relationship in his mind to a high school biologist's staining of a cell that's dividing, and he thought these look similar. What would happen if I put an electric field on E. coli cells growing in culture? And when he did that, he found in these electron micrographs that the little E. coli cells had grew into long filaments, and that was quite unusual. And he knew that agents that caused filamentous growth in bacteria had sometimes use in cancer treatment. He tried to get the National Cancer Institute to carry out some studies. They were completely uninterested because of the discovery in his lab shortly thereafter that the reason for the filamentous growth had nothing to do with the electric fields, but that a small amount of platinum that was used on the electrode to supply the fields had leached off and reacted with ammonium chloride in the buffer to form this very simple 11-atom molecule containing no carbon atoms whatsoever, cis-diamine dichloroplatinum 2, which subsequently became known as cisplatin. So Rosenberg carried out his own tests. He had never done this before, but it didn't take him long to figure out that he could take mice and implant a solid tumor in their ascites fluid, and in the control animals, they lived about 3 weeks before the tumor overwhelmed them and they died. Whereas in the test animal, a single injection of cisplatin, as it became called, after about a week, there was a necrosis that formed in the tumor. After about 2 weeks, it had grown small, and at the end of the third week, the animal groomed off the tumor and lived a normal life. So that was extraordinary. The NCI took it up. There was about a decade of clinical trials, and ultimately, in the late 70s, cisplatin became released for the treatment of head and neck, testicular, and ovarian cancer. And although it's not a new molecule, having been discovered first in the mid-19th century, it took a long time before one found any biological activity. The anti-tumor activity demonstrated shortly thereafter, approval by the FDA in 1978, and it was about this time that my lab, still working at Columbia, decided to get really interested because it looked like DNA was the target of this, and for reasons I don't have time to go into, we had done a lot of work on metal nucleic acid interactions. And then ultimately, in addition to cisplatin, two other Analogs were prepared carboplatin, where the chlorides are replaced by cyclobutane dicarboxylate, and oxaliplatin, where the amines are replaced by the RR isomer of 1,2-diaminocyclohexane. And we have oxalate here, known as oxalate for colorectal cancer. These molecules have become giants of cancer treatment to the point today where about half of all cancer patients worldwide getting chemotherapy are being treated with the platinum, which is really extraordinary. In the case of testicular cancer, it's essentially a cure. There are very few cures of cancer. There's about 9,000 cases in the United States each year, under 100 deaths. In fact, the statistical NCI booklet says 0 deaths. Lance Armstrong was the poster child, probably not quite as good a poster child as he was at one point. But nonetheless, he had stage 4 metastatic testicular cancer. It was in his brain, and he had essentially a complete cure. So how do they work? Well, there are 4 stages in the mechanism of cisplatin. One is entry, cell entry. I won't say much about it in detail, although I'll come back to that later. So you have to get your drug across the cell into the tumor environment. Inorganic chemistry shows that the chloride ions are lost in a stepwise fashion, ultimately forming this aquated form, which then goes to the most nucleophilic position in DNA, which are the N7s of guanine and adenine, and it makes specifically crosslinks, particularly the 1,2 intrastrand crosslink, which is a feature that conveyed by the cis geometry of the starting material. And then there's the cellular response, and there are thousands of papers published per year on the cellular response to cisplatin, and I'm going to talk just a little bit about that as we go on. So how is it that one knows that DNA is the cellular target, and what are the structures of the adducts? Well, one simple thing is that if you have a cell that's diminished in its ability to repair DNA, it's ultimately very sensitive to the hypersensitive to the platinum drug. And secondly, if you take patients who have been treated with platinum drugs, they have higher levels of platinum DNA crosslinks, which can be detected by antibodies on the buffy coat, or the so-called the white blood cells, which are the ones that have DNA. And it's the patients that respond that form and persist and have these adducts. And these adducts persist in blood 30 years after treatment of the patients who have been cured. So they don't have to be cleared from the body. But the connection between the formation of those adducts and a positive response is another reason, among very many, which I didn't list here, are actually thought to be the reason why DNA is the target. And so we began to look at the nature of the interaction of platinum with DNA. So DNA is a pretty complicated ligand, if you want to think of it from the inorganic chemist's point of view. But luckily, there's a high frequency of these intrastrand GG and AG crosslinks, with a smaller number of the 1,3 crosslinks, for example, 2 guanines with an intervening nucleotide. And then there's some small amount of interstrand crosslinks as well. And they are part of the story, but they're not the Grand Central Parkway, shall we say, of the story. Now, once it's on there, what is it about these adducts that leads to the selective death of the cancer cell? And the major thing, there's more than one thing that takes place, so it basically relates to DNA as a substrate for cellular processes, and that includes DNA synthesis, replication, and cell division, and transcription. And a key paper in my thinking was one that was published by Alan Eastman in 1988, demonstrating that cisplatin arrests the cell cycle at the G2M checkpoint. And it does that, the cell cycle is arrested so that the cell can repair the damage. And he concluded that the transcription of the mitotic apparatus necessary to cause cell division was abrogated, and I think that is actually what's happening. And if you want to read about it in more detail, there's a paper published with my graduate student Ryan Todd where we talk about transcription inhibition as a roadblock to RNA polymerases. But I'll give you a little bit of the data that demonstrates graphically how this happens. So in this experiment, which we carried out a while back, you have a coumarin and a fluorescein linked by a beta-lactam. And the coumarin will fluoresce, but because of the nearness of the fluorescein, as long as this connection is made, there will be fluorescence resonance energy transfer from the coumarin to the fluorescein. And so if you irradiate at 409, it will emit at 520, and the cells will look green. Now, the particular cells that we looked at had been engineered to carry a beta-lactamase, which cleaved this linkage. And so in the absence of something like platinum, once you put this substrate in there into the cells would actually fluoresce blue because the beta-lactamase would cut the connection between the coumarin and the fluorescein, and so it would be the coumarin that would fluoresce and you get a blue color. But now if you add platinum or any agent that blocked transcription, you would see a color turning green, and indeed this is what happened here. We had this green color that appeared, and that was proof that one has transcription inhibition. And in fact, I had an undergraduate at at MIT when I was there, when he was there, who prepared several thousand new platinum compounds using robotic synthesis one summer, and with the help of a postdoc, they screened those using this assay. And it turned out there were 4 hits, and all 4 of them had been taken by various companies into the clinic because they had profound anti-cancer activity. Now, another way you can look at it is to look at a site-specific cross-link. So along the way, we learned how to make site-specifically platinated DNA by synthesizing a short oligonucleotide that had a GG and only a single pair of Gs in, say, a dodecanucleotide, add platinum, separate and purify it by HPLC chromatography, and then ligate it to pieces that could be annealed, and in addition to ligate some additional piece with a small starter RNA, which I show in red here, which is labeled at the 5' end with a P32, and could be pulled down on a magnetic his tag specific magnetic bead. And then you bring these together, again, annealing and adding ligase, and so you have what's called an artificial transcription elongation complex. So this guy is ready to transcribe the RNA off of this leader sequence, and so if you now add the nucleoside triphosphates for RNA, you could do this transcription walking experiment. In the absence of any block, you'll go all the way down to and get 133 nucleotides. But if you have a good block to transcription, you'll just make 62 nucleotides. And then running an electrophoresis gel is quite easy to take a look and see whether your platinum compound could block transcription. And indeed, you could get 133 in a control experiment, but if you took cisplatin 1,2 intrastrand GG crosslink, the cisplatin 1,3 intrastrand GTG crosslink, the diaminocyclohexane platinum intrastrand GG crosslink, that is the oxaliplatin adduct made by oxaliplatin, and the 1,3 crosslink, and you look at the RNA that comes out, in every case, you get a complete block to transcription. And these compounds are active drugs, and in compounds that are not able to make this crosslink, not able to block transcription, even though they might modify DNA, you get no activity. So in cartoon form, what you can take away is the mechanism of cisplatin works because there is damage to the DNA. RNA polymerase II, the main transcribing DNA transcribing enzyme in the cell, is blocked. There's a stalling process which triggers many cellular processes, including DNA repair and apoptosis. So it's a battle between those two. And failure to recognize the damage in answer to this call is what you want in the cancer cell. And cancer cells are well known to be deficient in their ability to repair damage on their genome. They're busy doing lots of other things. It's not that they don't do it, they just don't do it as well. And in some work we did with Phil Sharp, we showed that the C-terminus becomes phosphorylated and ubiquitinated. What happens is then the repair enzymes come along, pull it away, and if the repair enzyme takes it out before apoptosis is triggered, then of course the damage will not affect the cell, and that's what you want to have happen in the healthy cells. There's a lot more to the story. I don't have time to go into it. I could tell you what our hypothesis is as to why testicular cancer is cured. It relates to this mechanism. I'll just say in very broad terms, it has to do with the fact that a unique form of a protein that we found a long time ago binds to the intrastrand GG crosslink as a result of a specific phenylalanine that inserts itself into the hydrophobic notch across from the GG crosslink. And in testicular cancer cells, that's abrogated, and that can't happen, and so you basically don't get the repair shielding that takes place in cells where that can happen. We could talk about that in the question period if people are interested. Now, of course, platinum is not a panacea for cancer. even though most people who have chemotherapy get a platinum compound. There are toxic side effects, and there's resistance, including acquired or inherent resistance. There are aggressive tumors, and there are cancer stem cells, which in my mind, and although this is still controversial, have a lot to do with why cancer recurs and why it metastasizes. And I'm going to come to that at the end of my presentation again. And so we and others have looked at a number of non-classical platinum compounds, including platinum-4 compounds, which we call dual threat, because they can bring other things in beside the platinum DNA adduct-forming chemical, and also monofunctional compounds. So let's talk about the strategy for delivering platinum compounds in vivo. And a lot of people have worked on this, including pharmaceutical companies. And nanotechnology and nanomedicine has been called to the fore in a number of areas. I think by no means has it reached its full potential, but these are various nanocarriers that have been used for cancer therapy. And one of the ones that we worked on early was the single-wall carbon nanotube, which we did in collaboration with Hongji Dai, Dick's colleague at Stanford. And I met him when I was on sabbatical out there, and we had a really nice interaction. The single-walled nanotube can be, we call them longboats, actually, and you can add oars to the longboats, like the Viking longboats, and their amino groups, which can be then added to a functionalized platinum compound, which is formed by taking the dihydroxo form of cisplatin. If you oxidize cisplatin with hydrogen peroxide, you make the trans-dihydroxo platinum-4 compound, and then you can, using chemistry that we heard about related to how Celanese made its compounds in the early days, you can link two species in the axial positions, and one of them can go to a tumor cell homing device, for example, folic acid. And so if you take this and put it in blood, it will go into tumor cells binding to the folate receptor, and then you have a prodrug. As the platinum compound gets taken inside the cell, the intracellular reducing agents will take off the axial ligands and restore cisplatin, and that's then the active pharmaceutical ingredient. Hongji sent us these functionalized longboats. I called him up and I said, how many amines have you got? We've measured how much platinum's on there. He said, we got 80. We measured 82, and I said, okay, we're in business. We can do it quantitatively. And then what you can also do is you can take those tumor cells that you treat, and you can look at the end of the day to see whether they have actually released the platinum drug and it has gone to the nucleus and made the intrastrand crosslink. And with the help of an antibody that we got from Jürgen Tamala, who is a co-author on this paper in Germany, we were able to show that that's an antibody specific for the cisdiamine platinum 1,2 intrastrand crosslink. So it's formed in that process. We then looked at the cytotoxicities in different cell lines using a particular assay. We looked at a human choriocarcinoma and a nasopharyngeal carcinoma, both with folate receptor positive, and then as a control, we used human testicular cancer cells, Entera, which are very sensitive to cisplatin, but do not contain the folate receptor. And we found the red mark is the single-walled carbon nanotube carrying molecule homing in on the tumor because of its folate receptor ability. You see, this is a survival versus concentration of platinum. You want the smallest number possible. So basically, 19 nanomolar concentration of the platinum nanotube in platinum was effective, whereas cisplatin took, considerably greater than that. Also for the KB cells, but not for the Entera-2 cells. So that was a nice proof of principle that you could get selective targeted delivery of this cytotoxic agent to cancer cells. And so we then went on and looked at nanoparticles, and here's where I began a fantastic collaboration with my colleague Bob Langer and Omid Farrokhzad. Now, Bob, of course, I'm sure most, if not all of you, have heard of, chemical engineering at MIT. And Omid was a postdoc in Bob's lab, is now a full professor at Harvard Medical School, and is a really wonderful nanoparticle scientist, but also entrepreneur. And I'll get to that in a few moments. Omid, Bob, and I have founded a company known as Blend Therapeutics to take our knowledge of platinum compounds to the clinic. And as I said, I'll get to that shortly. So you can take a polylactic coglycolic acid polymer, and then with the use of a platinum-4 prodrug, prepared by taking the dihydroxo and reacting with hexanoic anhydride. You can make the dihexanyl compound, and then that will go into the hydrophobic core of the nanoparticle and be carried inside the cell. And then these particular nanoparticles were decorated with aptamers on the outside that were specific for the prostate-specific membrane antigen, which of course is present in prostate cancer cells, but also present actually on many cancer cells. And we could then look at the platinum amount of drug loading that we saw and also the size distribution. And we could also visualize that when this nanoparticle got into cells, it released cisplatin and made the intrastrand crosslink on DNA in the nucleus, again using the same antibody that I showed previously. And when we looked at the cytotoxicity profiles of these nanoparticle-delivered molecules, if we had the prostate-specific membrane antigen present, we had a very effective compound. If we didn't have it present, it was less effective, and the targeted nanoparticles were 4-fold more effective against the PMSA cells and 84-fold more effective than cisplatin. Okay, that's cells. What about if you move another step forward and you look at animals? We measured the maximum tolerated dose in rats, significantly higher than that of cisplatin. We measured the blood retention in rats after an hour, 77% retention in blood versus 1.5% of cisplatin, most of which gets eliminated, excreted within the first minutes of injection. And then we looked at tumor sizes in a xenograft study, in the nude mouse xenograft study. The dose schedule is shown on the slide. And the bottom line is the platinum nanoparticles were more effective at curing cancer at a third the dose of cisplatin with no recurrence after 45 days. So that looked pretty interesting. Pretty easy to cure cancer in mice. Well, not pretty easy, but it's relatively easy. That doesn't mean it's going to work in humans. But it gave us a lot of confidence that this approach, this way of making a platinum drug have longer lifetime in blood and be carried specifically to cancer cells is a way that we might be able to make platinum compounds available to a much wider range of cancer treatment modalities than we have today. And I can tell you that the scientists at Blend have been absolutely spectacular. We were very fortunate in getting top-notch scientists from places like Merck and from SmithKline Glaxo come in and work with the company. The company was founded in January of 2011. I never founded any company before, unlike Bob, who has 40, and Omid, who has 3, 2 of which have gone public already. Blend is going in man next year. We have our FDA meeting going on now with its first therapeutic agent of a new platinum compound, which will be the first targeted cytotoxic, to the best of my knowledge, ever. So that I can't quite tell you what it is yet. I will be able to within a week or two. There's going to be a big announcement because the clinical candidate has been chosen. But I can just say that we'll be able to pick our patients with a genetic test, and it's just fantastic. It builds on our science, including a lot of which we didn't say, but it's essentially an invention of the company, and I'm very proud of these guys for having gotten it there. And I hope to be able to fulfill my long-lasting wish of curing at least one person of cancer before I leave this planet. All right, now let me tell you a little bit more about back to the research lab. You notice that everything has been built around a pair of cis leaving groups and making a cross-link, and of course organic mustards do that as well. And some interesting things came along as we were studying. Various people called me and talked about things, and one of them was Kathy Giacomini at the University of San Francisco Medical School. And Kathy was looking at the way in which oxaliplatin gets taken up into cells. And oxaliplatin has this 1,2-diaminocyclohexane, and it's got an organic unit, and she had thought that it might be taken up by the organic cation transporter. And so we began to work with her, and indeed it is taken up by the organic cation transporter. And so this is the cisplatin here. This is carboplatin. You see very little difference between cells which do or do not have the organic cation transporter. That's what this ratio is. And then you see oxaliplatin, and you can see a much greater degree of uptake by the organic cation transporter. And that's used as proof that, in fact, one has an uptake mechanism that is not available to the the first 2 platinum drugs. And then I thought to myself, well, you know, oxaliplatin is not a cation. In order for it to be a cation, it's got to lose oxalate. So what if you actually took a cationic platinum compound, and we had a number of them in my laboratory, and one of the ones we had was this one here, where one of the chlorides has been replaced by a pyridine ligand. And that compound was around because Steve Hollis, a former student of mine, worked in the early days, in earlier days at Engelhard Industries, which was one of 2 companies, Engelhard and Johnson Matthey, that set up small platinum drug internal discovery groups when cisplatin became a $1 billion drug, and they thought, well, can we cash in on this? Both of those were closed down. Hollis found this pyridine compound, and we worked with them on it and published, and it's effective. It kills tumors in the Rosenberg model, where you implant in the ascites fluid, etc. Now, remember the original structure function rules, or if you don't remember, I'll tell you, they were published in the early 70s based on attempts to try to get better agents than cisplatin. And they were as follows. First, the compound should be neutral. It should, the pyridine compound, I'm going to call it pyroplatin, is not. It's a cation. It should contain a pair of cis-leaving groups. Well, that doesn't. You only have 1 chloride, and we proved that the pyridine didn't go away, so it doesn't have that. The other 2 ligands play an important role. They should be inert and neutral. Well, that's true. They're 2 ammonias. But the reason that those rules came along was because the early attempts to make compounds which we call monofunctional, because they can only make 1 bond to DNA, included replacement of the chloride with an ammonia, so you have triamine chloroplatinum 2, or with a diethylene triamine, and those are completely inactive. And then the trans isomer, also completely inactive. So that's how those rules came about. Remember that the one that I showed you a few moments ago that's taken up by the organic cation transporter has a pyridine here. So I thought, well, how could that be? Is our mechanism wrong? I mean, how could that be active? Or does this thing go by a different mechanism? So we began to look at pyroplatin, which did form monoatics on DNA, violates those structure-activity relationships. What are the features that make that active? So the first thing we wanted to see was, what's the structure of pyroplatin on DNA? Now, it took my graduate student, Trisha Takahara, a wonderful student, a long time, years, to get crystals of the cisplatin DNA adduct, which we could study by X-ray crystallography, and this is the classic structure, intrastrand, cross-link in the major groove. It took Ryan Todd, graduate student, working with Katie Lovejoy, about 10 weeks to prepare, crystallize, and solve the structure of pyroplatin on DNA. It's a great tribute not only to their abilities, but also to the way in which the technology evolved over the following years. And you can see it's common garden variety Watson-Crick base pair DNA with platinum bonded to the N7 of guanine, because we put it there. We made this up. We made it that way, just hanging out there. So how could that compound possibly be effective? I mean, is the mechanism the same? Remember, I told you cisplatin inhibits transcription, so there's very little transcription bypass. It does get repaired by the nucleotide excision repair pathway. I didn't give you that before. That's on this axis. The platinum diethylene triamine compound, completely ineffective, that I showed you before. Very little transcription inhibition, big bar, and no repair because the cell doesn't care it's on there. But pyroplatin's like the baby bear of the papa bear, mama bear, baby bear triad. It's in between. It inhibits transcription, and it's not that well repaired. So maybe it's, in fact, going to be a reasonable species. So it inhibits transcription by Pol II nearly as efficiently as cisplatin, and it eludes nucleotide excision repair, the pathway, compared to cisplatin. Moreover, the cytotoxicity against different types of cells is completely different. So here's cisplatin, and this plot, which is a small portion of what's known as the NCI 60 screen for effective cancer drugs, you have a whole bunch of different types of cancer: colorectal, breast, colon, lung. And the mean IC50, the mean concentration that kills half the cells in culture, is the bar down the middle. Everything better for these types of tumors is on the right-hand side. Everything worse is on this side. Oxaliplatin has a very different profile, and it was on that basis that oxaliplatin was developed as a drug, the third platinum compound, and quickly became a $1 billion drug. And so we were excited to see that pyroplatin also has a very different spectrum of activity from either of these two. And I was at NCI on sabbatical in 2012 and saw also a very, very different spectrum across the whole NCI-60 screen. But look, the mean IC50 of the pyriplatin is less potent by an order of magnitude, more than an order of magnitude than cis or carbo, so we had to make it better. We want to keep the profile differential, but we want to make the toxicity better. So we needed to have a clue about how to go about doing that. Luckily, my former graduate student, Dong Wang, who had done his PhD thesis working on RNA polymerase in Roger Kornberg's lab, is now on the faculty at UC San Diego. And Dong and we collaborated on getting the structure of pyriplatin in the active site of RNA polymerase II, and we could see that it entered the site where you make the where the extending RNA strand could add the complement to a platinated guanine. So this is now, say, the Watson strand or the template DNA in blue. There's a little piece of the non-template or Crick strand in green. This is a particular part of the RNA polymerase structure, and this is the growing RNA strand. The pyridine causes the platinum to rotate about the N7 of guanine, putting hydrogen bonds here, and as a consequence of those interactions, that prevents translocation out of the active site, stalls the polymerase, and without making a cross-link, blocks the polymerase from extension. So we thought, well, we need to block it better, and we thought by looking at the structure, if we make the pyridine larger, we could do that. And so Ga-Young Park, my postdoc, actually made a small family of different heterocyclic amines, and when she got to the one that contained phenanthridine, it was a real winner. If we look at these different cell types, lung, ovarian, cervix, breast, etc., we see the cisplatin efficacy, the oxaliplatin efficacy, the pyriplatin, and the phenanthroplatin. The phenanthroplatin is much better than anything on there, and it's substantially more potent than cisplatin or oxaliplatin by factors of 7 to 40. So we figured out that we could take monofunctional adducts and make them effective, and I can tell you that that would be a really good thing for a pharmaceutical company that works on platinum compounds to think about taking forward to the clinic. Now, it did more than that, however, and that's why the title of this talk uses the word transition metals or something like that rather than platinum. It told us that we didn't have to limit ourselves to platinum or even necessarily to third-row transition metals. We had not looked at those earlier metals before because an intrastrand cross-link bends the DNA and cannot accommodate 2 additional ligands in the axial positions of a 6-coordinate molecule compared to a 4-coordinate molecule. But knowing that a monofunctional compound could be effective completely changed that, and so we began to look at osmium, rhenium, tantalum. We've made those all. And one of the ones made by Rama Sunderlingam, who's one of the best postdocs ever to have gone through my laboratory, went right from my lab to a tenured position at King's College London, found that this osmium nitrido trichlorophenanthridine compound in bathophenanthroline was quite effective as an anti-tumor agent. And I don't have time to take you through a lot of the details. Now we have much more powerful methodologies which are going to be shown on these slides without a lot of time for me to look at all the detail. But these are biomarkers related to DNA damage in the cell, related to checkpoints that occur as the cell is desperately trying to save itself from the chemical assault, and ultimately P53, which is the master molecule in the cell that distinguishes and divides in between repair pathways and cell death. And we could see the osmium in the nuclear DNA. One of the beauties of being inorganic chemists studying biology is you have the ability to look at a unique element in there. And then my colleague Matt Heeman has developed these heat plots which look at siRNAs to give you a general sense of where the damage is occurring. And long story short, we've published this, our first paper on the osmium compound, is that by G2M phase cycle arrest, similar to that as cisplatin, the phenanthroline compound acts by a cisplatin mechanism, whereas the bathophenanthroline goes to the ER and kills the cells by the unfolded protein response. Now, the last little bit I want to talk about are cancer stem cells. So cancer stem cells are a small population of the tumor cell environment which can self-renew, differentiate, and form secondary tumors. They can go to remote sites. They can trigger metastasis. And in order to get them, it's believed you got to get them in transit somehow or other. And it's hard to study them because you can't really get them pure. And my colleague Bob Weinberg, who I grew up with in Pittsburgh, I've known him forever, is one of the experts on this. So we got some cells from him and a lot of knowledge. And so if you look at the initial tumor, it's heterogeneous in many ways, including the stem cells. Current cancer treatments are such that typically the stem cells survive and the tumor recurs. The desired situation, of course, would be to knock out the stem cells as well as the cancer cells and get everything killed, which might lead to a durable response. And you can look at that website to see the difference. So the quest has been hampered by the ability to get those stem cell-rich populations, and you can do that by knocking out genes such as CDH1 or by treating the cancer cells with a non-cancer stem cell-specific compound like paclitaxel. And so we did that. We got some Hemmler breast cancer stem cells from the Weinberg lab, which had 5 to 8% cancer stem cells. Treat them with paclitaxel for 10 nanomolar for 4 days and enrich. We call those Hemmler tacs. And we can get up to about 54% cancer stem cells. And you can monitor the amount of cancer stem cells by looking at a particular marker on their surface known as CD44 using a fluorescently labeled antibody. And so if we look at those cells growing in culture and we look at what happens when you treat them with cisplatin, carboplatin, oxaliplatin, and another drug that almost made it but didn't through the FDA, which I will show you but not talk about. This is a histogram essentially that shows you the percent of cancer stem cells, and you see that what happens when you treat with the conventional drugs, you enrich in the cancer stem cells. You don't want that to happen. So when people are getting treated, if the treatment is not effective, the tumor could become much more virulent, and that does happen. There's no question about it. But if you take the Hemmler tac cells, which have been treated with a taxane, you can increase those in the stem cells from 7% to 33.9%, and now you add these same 4 agents, no change. So it's not hitting the stem cells at all. On the other hand, if you take the Hemmler cells, you take the Hemmler tacs, you see you increase them in stem cells, and now treat them with this osmium compound, you knock it from 53% down to 5%. There's no rationale whatsoever behind that. It's plain dumb luck, but it happens. And we were incredibly excited to see that happen. It's selectively toxic for cancer stem cells. And the selectivity factor is 2.31, the enriched versus the depleted. And that compares with numbers that Bob Weinberg obtained early on in screening a library, chemical library, for compounds that did the same sort of thing. And his best 2 compounds were selinomycin, 8.45, and abamectin, 2.26, similar to what we have. And a company was founded by Weinberg and colleagues to pursue that. You can also look at something related to tumor recurrence. The ability of the stem cells to self-renew comes from their ability to make these so-called mammospheres. I don't have time to take you through a lot of that left. And you can see that the osmium nitrito compound inhibits the clonogenic growth of those. And using an RNA signature assay, which Mike Heeman developed, we could see that compound 1, which is that osmium compound, is positioned in a principal component analysis of these RNA signatures. I realize that's a red, white, and blue box, not a black box for you, to be able to attack both the nuclear and the ER parts of the pathway that lead to cell death. And we think that's why it's so specifically effective, and we hope to be in animals with that osmium compound in the very near future. So, to summarize, I think from understanding we've moved to knowledge, and from knowledge we've moved to improvement. And I think that's something that you want to do in life. Hopefully the stem cell attack will turn out to be something really valuable. We'll find out, but it's an area that we're now pursuing quite vigorously. But I want to finish with a retrospective. Now, this is a wonderful device here, those of you who are speaking after me. It's a timer, and I have 20 seconds left. And I might take a minute and 20 seconds, if you excuse me. But I always wondered, as we were working through this, in a retrospective sense, that if we took one of our new compounds, like phenanthroplatin, and went back to the original Rosenberg experiment, and we went to E. coli, and we took E. coli cells growing in culture, would those newly active compounds produce filamentous growth in E. coli if we could run the clock back? So this is the Rosenberg experiment. This is a scanning electron microscope image of the untreated and cisplatin-treated cells. So we decided to look at cisplatin, and then the monofunctional triamine chloroplatinum cation, which I told you was not effective. The pyroplatin, which had a very different spectrum of activity, but wasn't that active, but it was somewhat effective. And then finally phenanthroplatin, which retains that great spectrum of activity, but is super active. What happens if we go back to Rosenberg, run the clock back? So now I'm going to show you micrographs. These are not electron micrographs. These are light micrographs, so they're going to look different, but I think you'll get the picture. These are E. coli cells growing in culture. They look fine. We treat with cisplatin, and guess what? Rosenberg was right. They go into filamentous growth. And then we treat with the triamine platinum chloride compound, chloroplatinum triamine, and you don't see any filaments because there aren't any. It's not active. And then we treat with pyroplatin, and if you look closely, there's a couple of these little guys that are trying to become filaments. They're teenagers, not adults. And then finally, phenanthroplatin treatment in filamentous growth. I just thought that was cool. I don't know whether it means a thing, but if science isn't fun, you shouldn't do it. So I'm going to stop by thanking my group members, whose names are shown here. Not everybody I was able to talk about. My collaborators who've been great in helping. And then just the latest members of the platinum subgroup. I mentioned some of them along the way, and I want to thank them. They've been fantastic. MIT is a great place to work, and we get really great people. I thank you for your attention, and I hope I've left a little time for some questions.
Questions, comments?
So I'm glad that to see that you expanded into other transition metals, because one of the things that I always wondered is why platinum? I mean, you showed that there, of course, was this serendipitous experiment, and so of course people follow up on serendipity. I also wonder if it has to do a lot with the fact that platinum is really a magic element in many ways, because it's a tremendous catalyst that seems to balance, not overreacting, so having sort of just the right amount of reactivity. Can you speak a little bit to your thoughts about the role? I mean, from not. I mean, we know platinum is special, but what are the prospects for moving beyond platinum? And just a little bit more about your thoughts about platinum.
I'm happy to do that, and I would ordinarily I might start by saying that's a great question. So when somebody says to you that's a great question, it means one thing: they know the answer. So think about that. Okay, so we asked that question, and in fact, we looked at other compounds. I got some support from industrial sources, 20 years ago to do this. If you just think, what about above platinum in the periodic table? So palladium would be the obvious one. The problem is that many of the early transition metals, lighter transition metals, are kinetically more labile. And so they tend to go to the thermodynamic sinks in the cell and get stuck there and cause side effects and be toxic. Whereas platinum has just about the right kinetic properties to let its chlorides go. We measured the half-life in cells by platinum-195 NMR spectroscopy. That's another whole story, a long time ago. And the half-life is 2 hours to lose the first chloride. And you get no reactivity until you lose that first chloride ion. And so that's, and that, of course, is I can expound on this for a long time, but the major difference between inorganic chemistry and organic chemistry is that organic chemists are largely under kinetic control, otherwise it would be CO2 and ammonia and water, whereas, inorganic chemists are under thermodynamic control because of the rapid metal-ligand exchange rates. In terms of other metals, now I think that the door is open. Now, we were by no means the first to look at something other than platinum. and get it into the clinic. There are ruthenium compounds in phase 3 trials in Europe. So ruthenium is right above osmium in the periodic table. I don't know whether they've ever tested them against cancer stem cells, but our paper that I showed you at the end has just come out like a few weeks ago, and I can't imagine they aren't going to do that pretty soon. So I think that there is room for expansion. I think that hopefully that with blend and other compounds that the pharmaceutical industry will finally realize that they should put some effort into this by supporting it. They don't do any research anymore, but they could support it. And that there's a real hope for metals in medicine being much more effective, not just for cancer, but all sorts of other diseases.
The folate or the nanoparticle delivery processes, is that normalized through the various platinums that you've been talking about? Do we know independently of the delivery mechanism how active the various compounds are?
Yeah, so the compounds used in the clinic, we don't know too much about. Even with cisplatin, the actual delivery is a little bit controversial. It was discovered not long ago, meaning within 5 years or so, I think, that cisplatin can go in through the copper transporter. And in a way, that's not too surprising because copper is taken in by a sulfur-containing ligand methionine in that transporter. And the so-called hard-soft acid-base theory in inorganic chemistry tells you that platinum would want to bind there too. And so the question then is, when it gets through that transporter, is that going to be active or is it going to block it? And then you have exporters which take copper out of the cells, which work by a similar mechanism, and they could be taking platinum out and causing the cells to become resistant. So that's an ongoing story which we haven't been involved much in. There's also some degree of passive transport, and it's a question as to whether that's even fair enough, whether there is such a thing at all. But a lipophilic compound could get through, and so the business of getting things across the cell membrane is not simple, and there's not a simple, well-defined answer to that. Not something we've put a lot of effort onto, other than the work with nanoparticles. That we do know how they get in.
Hi. Yeah, it was a great talk. So I was wondering, the number of nucleophiles that DNA offers is probably much smaller than the number of nucleophiles offered by protein or RNA in the cell. Is it likely that these compounds are reacting with many other nucleophiles, but the reactivity with DNA is much more permanent or much more profound, and this is why you see these drastic effects? And what can you comment on reactivity with proteins, for example, or RNA?
Yeah, so RNA would be similar to DNA, except that you have some tertiary structure which might be a little more difficult to penetrate. But one can penetrate, especially if you have an intercalative type of unit. We found metallointercalation and named it a long time ago, and it does go after things like sulfur, even in tRNAs, which have a fourth IOU. There's no question that there's lots of other places that platinum can and will interact with in cells, including proteins. There's a lot of counterculture out there. People think that the real mechanism involves RNA, not DNA, or the real mechanism involves a specific protein. And I'm open to, I'm completely devoted to the truth. And so we teach our students that you can never prove a mechanism. You can only disprove it. And so if someone comes along and can show that our mechanism for how cisplatin works, there's something better that can explain all the data in there that's out there, I will cede the floor and say, thank you for letting me leave the planet with some knowledge that I didn't have. But in fact, just the opposite has happened. Okay, so cancer biologists, people who are doing drug development, people who are doing research, they use platinum compounds, and they discover that if they think of RNA and transcription and the nucleus as being the key elements of the mechanism, they can then explain the data that they have as to why this particular species added or compound added in addition to platinum makes things more effective or less effective and so forth. So the answer is yes, there's no question. There's as my colleague John Essigman would say, a dog's breakfast of molecules in the cell, waiting for cisplatin to come along. But I think the main focus needs to be on on the nucleus, on DNA, and on the processes of transcription inhibition versus repair. I mean, is it oversimplified? Maybe, but it seems to be explaining all the data as we know it.
Hi, over here. This way. I'll stand up. It makes it easier.
Oh, thank you.
Very nice talk. I was a bit confused with the cisplatin reaction with the cancer stem cells. So it was increasing cancer stem cells, but it still has a 90% rate of cure for prostate cancer. So is there a recovery?
Testicular cancer.
Testicular cancer. So are those patients then seeing recurrence in later years, or is it just latent and then doesn't come back at all?
That's a good question. I mean, there is very little, to the best of my knowledge, and I stress that I'm not a clinician, cancer recurrence in cisplatin-treated patients, and usually it's thought to be a second primary tumor and not a result of the original tumor. Whether it's a result of the platinum or not, I mean, that's really difficult to prove. 30 years later, their people were perfectly fine, and they carry the platinum on their cells. I think the answer, is that is so effective that even if the stem cells get out and create a secondary metastasis, I mean, Armstrong, stage 4 metastatic in the brain, that genetic image or fingerprint is there in the brain cells. It's not going to be absolutely identical, but it's testicular cancer in brain, and the pathologist can see that. So it's going to knock that out if it's going to knock it out in the primary tumor, right? So even if the stem cells, and they did, something got there, metastatic cancer occurred in Armstrong and lots of people, and that's how they die. They don't die from the initial tumor. Whatever is going on that's causing it to be effective is going to be carried out there. What you'd like to do is be able to knock out stem cells. Then you don't have to have a mechanism that's tumor type specific, because it could knock out perhaps all stem cells. Those are the little kids that are undifferentiated. And there's reason to believe, I don't know that we know the answer to this from the genetics, that there's differences. There's reason to believe, or at least I've heard, I'm not a stem cell expert, that they carry a lot of the same things from different tumors. They're very similar types of cells in terms of their origin and their genetic makeup.
Another question? To me, it's thrilling to see chemistry in the service of human health. This is a wonderful way for us to be treated by Professor Steve Lippard to the start of our symposium. Thank you again.
Thanks a lot, Dick.